All right, good morning everyone. My name is Chacon. I'm a senior here at St. Thomas University. This summer I had the opportunity to work with Dr. Plunkin and Dr. Tapanes Castillo on a project that involved the classification of adult zebrafish primary brainstem culture cells based on their molecular profile and cellular phenotype. The exploration of the topic of spinal cord injury can be approached by studying the different mechanisms that a species have to recover post-injury. In the case of the adult zebrafish, it has been used as a very useful model for axonal regeneration following a spinal cord injury. In our lab, we have previously established primary brainstem cultures to study this regenerative response in vitro. My role this summer was to further classify these brainstem cultures using immunocytochemistry methodologies in order to be able to classify them according to molecular profile and cellular phenotype. So the research question for my experiment is, what cell populations are present in brainstem cultures derived from adult zebrafish? In order to answer that question, we develop a hypothesis that our primary neuronal brainstem cultures contain different types of cells, including neurons, glia, and neural progenitor or stem cells. In order to molecularly characterize a cell, it is important to understand first its anatomy. As we can observe in this micrograph, a neuron comprises of three main distinctive features. First of all, we have the cell body, or soma. And in this part of the neuron is where the nucleus of the cell is located, and in this nucleus is where the DNA of the cell is stored. We also have the major process, which protrudes from the cell body and matures into an axon. This axon is the one that sends signals to other neurons. Finally, we have what are called minor processes, or cellular extensions that come from the cell body and they mature into dendrites. These dendrites have the ability to send signals, to receive signals to other neurons. In addition to neurons, we have in the central nervous system other type of nerve cells called glial cells. Glial cells have the main role of supporting these neurons by, for instance, in the case of the astrocyte, providing the neuron with nutrients, and in the case of oligodendrocytes, they are able to produce a myelin sheath to insulate the axon and therefore allowing the faster propagation of the electrical impulse. In order to talk about them further, like at the molecular level, it is important to first understand what the procedure of immunocytochemistry is. Immunocytochemistry is a molecular biology technique that uses antibodies to be able to label proteins located in and on the cell. Immunocytochemistry, it uses the antibodies. And in our lab, we would use a two-step fluorescent method, which I'm going to explain by this animation. We have a protein of interest, which can also be called an antigen. We have a primary antibody, which binds to this protein of interest, and afterwards we have a secondary antibody, which contains a fluorescent tag. What this fluorescent tag is going to do is that it's going to be able to allow us to visualize the cell under the microscope. Some of the antibodies that we use in our experiments are the anti-GAF43, anti-relating to antibody, and GAF43 relating to growth-associated protein 43. This antibody is used as a marker for axonal growth. We then have anti-PCNA, which is a marker for cell proliferation, and PCNA stands for proliferating cell nuclear antigen. Finally, we use the antibody nestin as a marker to locate stem cells in our cultures. As we can observe in this micrograph, GAF43 is being expressed in our cultures. DAPI labels the cells, the nuclei of the cells blue, while GAF43 is exhibited as green coloration. GAF43 is an intracellular protein found exclusively in the central and peripheral nervous system of animals. And in our cultures, we can say that we have a strong signal of GAF43 indicating axonal growth. We were also able to study the cell proliferation in our cultures by using the antibody against PCNA. And as I mentioned before, pCNA stands for proliferating cell nuclear antigen. pCNA is expressed in mitotically active cells throughout the cell cycle. And in our cells, in our 7 days in vitro cultures, we were able to identify pCNA positive and pCNA negative cells. pCNA positive cells more likely correspond to immature cells and like precursor cells, and also can represent glial cells. Now, pCNA negative cells more likely represent differentiating neurons. In this micrograph, we can compare these two cell types here. Both of them are pCNA positive, but they mainly show a difference in phenotype. For instance, this flat shape will be representing a mature type of glial cell, while this one will represent a precursor cell, and that's why they're very different. Now I would like to discuss the relationship between nestin and neural stem cells. Nestin is an antibody. We use the nestin antibody to locate stem cells in our cultures. In a stem cell, the basic definition of a stem cell is a single cell that can either replicate itself or it can differentiate into different cell types such as neurons or glial cells such as astrocytes and oligodendrocytes. Some of the characteristics specifically to the nestin protein is an intermediate filament expressed in the stem cells of the central nervous system. We use nestin to locate stem cells in our culture, and nestin expression is limited to precursor cells and not mature cells. Finally, we use a commercially available antibody that was raised against the zebrafish nestin. We can now observe a 3-channel micrograph, each indicating different features of our culture. We have in the first channel DAPI staining the nuclei of the cell. In our second channel, we have the protein tubulin being expressed in our cultures. Tubulin is a microtubule component that is expressed in the cytoskeleton and the processes of cells. Now, on the third channel, we have located nestin positive cells. And I would like to say this is the first time that nestin positive cells have been identified in brainstem cultures of adult zebrafish. Finally, on the last channel, we have a 3-channel merge, which indicates different features. So if we look closely to this last channel, we can identify the nucleus being labeled by the color blue, tubulin by green, and nestin as red. We can identify different populations of cells in this micrograph. For instance, we have beta tubulin positive, nestin negative type of cells. And we can also have nestin positive cells, beta tubulin negative. Now the question arises, how come a cell can be beta tubulin negative? And the answer to this, it will be that the tubulin is highly expressed in mature cells. So if this cell lacks the expression of tubulin, that can represent that this cell is a precursor or immature cell. Finally, we can identify also cells that express both tubulin and nestin, and that's why they have the yellow coloration. Some of the conclusions of our experiment, we used growth-associated protein 43 of GAF43 that shows strong signals in nuclei and neuronal processes. We were also identified the proliferating cells by the usage of the pCNA antibody, and we used the nestin antibody, and its effect, its expression suggests that the presence of stem cells in our cultures. Some of the future directions for our experiment is to continue characterizing these brainstem cultures by using double and triple labeling methodologies with the usage of additional antibodies. These are my references, and I would like to acknowledge definitely Dr. Plunkett and Dr. Tapanes Castillo for their tremendous support this summer. I would like to thank also Franz and Mem and my lab mates, as well as the School of Science, Technology, and Engineering Management, the U.S. Department of Defense, and the Minority Science and Engineering Program grant for providing us with this amazing opportunity. Thank you very much.
Any questions?
Yes.
You just
mentioned that for the first time you have found nesting and stem cells in the brainstem cells in zebrafish. What is the significance of that? Why is that important?
Okay, basically we want to study the, like I mentioned, the capability of zebrafish to regenerate their axons. So if we're able to identify nesting positive cells, that could be an indication that stem cells are present in our culture. And if stem cells are present in our culture, we can say that regeneration can be occurred. So what we do is that, as I mentioned in the future direction, we would like to study, use doubling and triple labeling to study the different interactions of nesting with other antibodies. Yes?
Is there any threshold value that you might identify in the number of cells that you're counting there that might indicate that there is important for the growth of axons? So you're looking for 3 different cells,
Yes.
but there's the qualitative description. And now, if we're thinking about the quantitative part, how much of each of those families of cells you need to guarantee a continuous growth? Can you say what the threshold value for them, or do you just want to see are there those cells, or it's more important to see how many of them could be there?
Well, that's mainly the way we can do it. Instead of getting an actual number of counting the cell, we can use different antibodies, right? The antibodies, the combination of antibodies will confirm the different expressions and the different functions of that type of cells. So let me just go back to show you, for instance. For instance, we can have a cell, in this case, that is nesting positive and tubulin negative. So we're using different antibodies to be able to explain the function of this cell in this culture. So if we are able to use double and triple labeling antibodies, we can locate cells that have various functions, and that can contribute to the regeneration process.
But the number of them, it doesn't matter the size, how many of them you have there.
Well, the thing is that, as mentioned, Rachel presented, we have different types of cells. We have 7 days, we have 14 days, sometimes even 20-day cultures. So in terms of the size, as time processes, they grow older. But if we're able to count more nesting positive cells, we can then say that more regeneration is taking place. Any other questions? Yes.
You're talking about an antibody to showing a expression.
Yes.
You just now mentioned function, proliferation, and so on. Are there actually any other ways to show the function of such a protein?
A function of such a protein.
Like you were talking about proliferation, and you're just actually showing that by doing a staining of a protein. But that's just an expression. It doesn't mean anything that it's actually doing something. It's showing that it's there. Are there any other possibilities to show a function?
Well, yeah, actually, like the first study, since we're doing in vitro studies, we can then use the antibodies, right, that to identify the different properties of the cell. We can use them in vivo cultures and study their expression and to see how these type of cells behave. Did I answer your question?
No,
Yes.
we can talk about it.
Is the lab doing in vivo culture?
In vitro, yes, in vitro.
No, in vivo.
In vivo, no. Another group is going to explain their results in terms of in vivo cultures.
And could you go back to your reference panel for a moment, please?
Okay. Yes.
I just want to see that. So,
Okay.
anyone has any other questions?
I have one question, but it's twofold. Can you tell me what myelin is made of?
The myelin? Well, I don't know exactly the composition of myelin, but I really, the only thing I know is like the function that allows to insulate the axons. I know it's a protein, but I don't know exactly the exact component.
Okay. And my second question is, you showed the nice, at the beginning about the primary and secondary antibody, you showed a nice demo there, you know.
Yes.
Why 2 antibodies? Could you use one?
The thing is that in order to visualize the cells, we need a secondary antibody that has the fluorescent type. there is no primary antibody that has the coloration, that can provide the coloration for the cultures. And there are other type of experiments that actually use more than two, so we can have a stronger signal and identify better the expression of the antiproteins.
If there's no more questions, one more.
You get a approximation of the concentration of protein by the intensity of the signal, of the fluorescent signal? Do you think you can get it?
Well, yes, I assume we can make a relationship between if the protein is expressed in high amounts, like tubulin in mature cells, we can say there is a high concentration of proteins. Like cells mature, so they become bigger and then they express more proteins if they have it on their cell.
Okay. Thank you very much.